Hello everyone, this is the video on the challenging topics on nervous system and nervous tissue, chapter 11 of the Marieb textbook. So we're going to start with just quickly going over the nervous system functions as a broad. They are a master control and communication system for the body. The function is to detect any changes, process this information, decide and perform actions if required, right? So let's look at the divisions. The nervous system can be broadly divided into central nervous system and peripheral nervous system. Central nervous system is our brain and spinal cord found in the dorsal cavity of our body, and that is the integrative and control centers. Whereas peripheral system is everything outside of this, right? So we have our cranial nerves, 12 pairs of nerves that arise and go to our brain. And then spinal nerves were 31 pairs, which arise and go, take information to our spinal cord. So they're the communication between the rest of the body and the central nervous system. We can further divide the peripheral nervous system into two portions in terms of the functional division. So sensory, afferent with an A, motor, efferent with an E. Sensory information will be received from outside of our bodies called soma, within our body called viscera. Visceral sensory neurons will bring information. From example, in this picture it shows the stomach. When we consume food, when it becomes full, it will send information to our brain, right? So this will, a sensory portion will take information to the central nervous system. Motor will bring information from the nervous system, central nervous system, to the periphery and to the effectors. Effectors are muscles and or glands. This can further be divided into whether we can voluntarily control or involuntarily control, right? So the voluntary controlling branch would be called the somatic nervous system. That's going to be the skeletal muscles, right? Whereas the involuntary or called autonomic or our visceral nervous system, that's going to be to our visceral organs. Okay, so it'll be to it will be to smooth cardiac muscles and to glands. This autonomic nervous system can be further divided into either parasympathetic or sympathetic. They are antagonistic to each other, which means they oppose each other. If one of them increases some activity, the other one generally decreases that activity, right? For example, sympathetic will increase our heart rate, while parasympathetic decreases our heart rate. Sympathetic is also known as fight or flight, while parasympathetic is known as rest and digest. So our digestive organs, urogenital organs, the activity will be increased during parasympathetic. Okay. Next, we're going to talk about the histology of nervous tissue. So two main functional groups of cells, neurons and neuroglia. The neurons will conduct nerve impulses while the neuroglia do not. Okay, neuroglia are support cells. They protect the neurons and they do not transmit nerve impulses. They are capable of cell division. Once we reach adult size, neurons do not divide, so we've gotten our maximum number of neurons by then, but neuroglial cells do divide continually. We'll talk about four central nervous system neuroglia, neuroglial cells, and two in the peripheral nervous system. So let's start with the central nervous system. The first ones we're going to talk about astrocytes. They are the most numerous in our central nervous system. They will help transport material from our blood vessels to neurons. So they help maintain a chemical environment, right? So this is blood vessel, capillary. The yellow is the neuron. So these are the cells that help to control what goes from the blood vessel to our neurons and vice versa. These cells will also be responsible for forming the blood-brain barrier, which we'll talk about in chapter 12. The next ones we're going to talk about is the microglial cells. These are smallest of the neuroglial cells. They are like immune cells of our brain, right? So they are phagocytic. They will swallow up any microorganisms or any cell debris. So this is the cell with multiple processes, right? Next, we're going to talk about the ependymal cells. Ependymal cells, hopefully by looking at the picture, you can see them. They look like cuboidal, columnar kind of epithelial cells, similar, but they have cilia. So they are found in cavities within our brain, 4 cavities which are called ventricles, and then also within the central canal of our spinal cord. Within these spaces, we have this fluid, CSF, right? So they help move the CSF within these spaces. They also, at certain spots within our ventricles called choroid plexus, they will also produce CSF. Again, we will talk about this in chapter 12, the central nervous system chapter as well. The last one of the central nervous system cells we're going to look at are oligodendrocytes. These are responsible for producing myelin. Okay, so myelin, which I actually have a slide shortly, is made up of mainly fat, right? So we'll talk about it in a second. So they will wrap around the axons and will insulate them. The first of the peripheral nervous system microglial cells we'll talk about are the satellite cells. Where they are found are actually surrounding the cell bodies of the neurons, right? And we'll talk about cell bodies. That's where they are the largest part of the neuron, and they contain most of the organelles, including the nucleus, right? So they will surround it. Their function is to control the chemical environment. So they control what goes in from, and to from the blood vessels and neurons and backwards, right? So they're similar to, hopefully you've already thought about it, it's the astrocytes in the central nervous system, satellite cells to the similar function in our peripheral nervous system. Schwann cells perform the same function as the oligodendrocytes in the central nervous system. They will produce myelin. So this is a nice image. You can see the axon. Okay, so the yellow is the axon. These are the Schwann cells. They keep wrapping themselves around, coiling themselves, right? They squeeze the cytoplasm out. So what's left is basically what was their plasma membrane. Hopefully recall that our plasma membrane is made up predominantly of phospholipids. So myelin sheath is basically made up of fat, right? So why myelin? It actually forms the white matter, which we'll talk about in a second. So it insulates our axons and increases the speed of conduction of those nerve impulses, right? In certain spots on the axon, we do not have any myelin. They're called gaps in myelin. It is also the term is nodes of Ranvier. Nodes of Ranvier. Okay, sorry, it says nodes of Ranvier. It's myelin gaps or nodes of Ranvier. So this we'll talk about later in this chapter, what the significance of these are. So multiple sclerosis, you may have heard of the illness, you may have known a little bit about them. So they are an autoimmune disease. Autoimmunity is when our own body attacks the own cells, right? Our immune system will attack our own cells. So here in this case, they attack the myelin, okay? So when they attack the myelin, so the myelin's lost temporarily initially, then the electrical impulses do not pass through, so they're not these individuals are not able to control the muscles. They have muscle weakness and loss, right? The Schwann cells builds up the myelin again, and they are asymptomatic for a little while. Then the immune system continues to fight against the myelin. Symptoms appear, so it's relapsing, right? Usually the immune system does become more aggressive. It does lead to more and more damage, but thankfully medical advances have advanced so well that it is now a chronic illness rather than a illness which took away the person within less than 10 years, right? So now they're living 40, 50 years without a lot of issues, okay? Long-term control. So white matter and gray matter. So in our central nervous system, you're going to find parts that look white in color. Those are containing predominantly myelinated fibers, myelinated axons, right? While gray matter contains mostly unmyelinated fibers and cell bodies of neurons. So let's look at those neurons, right? So this is a beautiful electron microscope image. You can see a very large portion of this neuron and then very tiny, thinner processes. So this large part is the cell body. It contains most of the organelles, including the nucleus, right? And then we have dendrites, short processes. The function of them is to receive nerve impulses. Okay, so they receive nerve impulses. Please remember that neurons are amitotic. They do not divide. Okay, once we reached our adult size, they have a high metabolic rate, so they are highly dependent on oxygen and glucose, and when that supply is cut, cells begin to die, basically. So axon is the other process. It's generally long and just one most often. That the function is to take the impulse away from the cell body. Dendrites bring it to the cell body, that's the receiving side, whereas the conducting side will be the axon. Around the axon is where we have, in the peripheral nervous system, Schwann cells. Schwann cells produce myelin, right? So nodes of Ranvier are these spots in the axon where we do not have any myelin. Okay, and then at the end of the cell, end of this neuron, it will not be one end. It actually will form terminal branches. Will branch out, axon terminals. That's where the axon ends. This is where, when we talked about the motor unit in the last chapter, they are able to innervate multiple cells, right? So next we're going to talk about neuron classification. We have two classifications, structural and functional, similar to joints, but here structural is in reference to how many processes, how many of those branches we have attached to the cell body. So if we have 3 or more branches, 3 or more of these processes, we call them multipolar neuron. Multipolar neurons are the most numerous in our body, right? They're found within the central nervous system. Pretty much all of the cells there are multipolar, and all of the motor neurons are also multipolar. So the other two are basically both for sensory. Okay, so if I said bipolar, hopefully would have said 2. So they're going to have 2 processes. One side will be the receiving end, so dendrite. The other side will be axon, the conductive end, right? These are found in special sense neurons. So our 5 special senses are sight, smell, taste, hearing, and equilibrium. Okay, those are our special senses. Those places is where we would find bipolar neurons. Unipolar neurons, again, if there was ever a picture in any quiz or any textbook, you want to pay attention to the cell body. If you see there's only one process attached here, you can see there are 2. So you'd be incorrect if you think about this as being 2 processes. It's basically one process that is split. There is a peripheral end and a central end because we have information coming from one side, going to the cell body, and then going to the other side, right? These are again sensory neurons. These are for general sense, right? So sense receptors found throughout our body, like touch receptors. Those are tactile receptors. Okay. Then functional, hopefully you can come up with the first 2, right? Sensory is information that's coming to our brain, bringing information regarding any changes in the environment, sensory information. Motor will be what's coming from the central nervous system to the periphery for the effectors. While interneurons, those are the intermediate connections which connect cells, neurons between within our central nervous system. These are found in our central nervous system. They are all multipolar polar neurons. Okay, so next we're going to talk about membrane potential. So we mentioned that the neurons are highly excitable. What that means is they will be able to conduct exchange ions and conduct information, right? Carry information. So the main one we're going to look about in terms of nerve impulse, it's called an action potential. Action potential is the same. The shape of the graph is exactly the same, irrelevant to the stimulus. Either we have it or not, and I'll go back to it and say that it's all or none phenomenon. Just before that, we need to talk about the different types of channels that we can understand. So you may have covered this in other classes. So leak channels, you can think of a leak channel is something that's all leaking, right? So this channel's open always. So as long as there's an electrochemical gradient, ions, particles will move in and out of the cell, right? Depending on which way is down the concentration gradient. Gated channels, this means, as the name suggests, they have a gate. They are open or closed only when a certain condition is met. And those conditions can be of three types. One is called a ligand gated. Ligands are chemicals. So our neurotransmitters, for example, acetylcholine is a ligand, right? So this chemical binds to the receptor, binds to this channel, it opens, right? Then voltage gated, that is sensitive to changes in the ion concentrated, in the charges, right? So if it goes, and we'll be talking about this in action potential, when it goes from -70 to -55, that change in voltage causes certain channels to open and close. Okay, the last type is called mechanically gated. You can think of somebody actually mechanically opening the gate. So a touch receptors would have mechanically gated channels. So let's first talk about membrane potential. So this is the charge difference between the inside of the cell and the outside of the cell. It's measured in voltage, right? So here is a voltmeter. They've got two probes. One is on the outside of the cell, one's on the inside. The difference is the membrane potential. And most of our cells have a membrane potential of -70. It can vary from different cells in our body, anywhere between -40 to -90 would be resting membrane potential. When this charge changes, we say it's carrying a current. Okay, this happens using ion channels, and it happens because of electrochemical gradient. So concentration gradient talks about the chemical gradient. Electro is about the charge. So positive charge will move towards a negatively charged area, and vice versa, right? So just before we talk a little bit more about this resting membrane potential, let's also look at some ratios, really. So I don't expect us to remember the numbers from this class. You will talk about this in subsequent classes, but what is very important for us to remember is the difference in the ion concentration outside of the cell and inside of the cell. There's quite a few ones that you want to remember. Sodium is number one, right? Sodium, there's a lot more outside of the cell. So the red and blue are blood plasma and interstitial fluid. That's the outside of the cell. The yellow is intracellular fluid. So we have less sodium inside the cell, very large amount outside the cell. It's the opposite in potassium. We have a high inside the cell, low outside. Calcium, where we came across in the neuromuscular junction, I mentioned it's low outside, but it's virtually zero inside, so it's still higher on the outside, so it'll want to move in. The next one I want to concentrate is the bicarbonate ions. Negatively charged ions, again, are higher on the outside. Next one is the chloride. Sodium and chloride are best buddies. So wherever sodium's higher, you can expect chloride to be higher, right? So chloride ions are higher on the outside compared to the inside. inside we have phosphate ions, which are higher, right? So they can be part of molecules, ATP, ADP, DNA, RNA, all have phosphate groups, and they all also have others with it. The next one is the protein anions. What I really want you to concentrate is this larger block inside. What is significant is that these ions, so negatively charged protein molecules, are very large. They will not move across the membrane. Okay, so there are three big reasons why we have a negative membrane potential. So first you want to remember is there's a higher extracellular fluid concentration of sodium, a higher intracellular fluid concentration of potassium. So three reasons why we have a negative membrane potential. One, the membrane is impermeable to those large anionic proteins. So anionic means negative charge. So if you have more negatively charged these protein ions inside the cell, the inside of our cell is going to be more negative. Another reason is that potassium is 25 times more permeable than sodium. So there's more leak channels for them. So we will have both of these are equally charged, right? +1 sodium, +1 potassium. So we will have more potassium leaking out because we have a higher intracellular concentration of potassium. So more potassium ions leak out compared to the number of sodium ions come in. So a few plus charges come in, more plus charges leave. So a net loss of plus charge, which means the inside is more negative. The third reason has to do with the sodium potassium pump, right? It pumps against concentration gradient. It likes this first few two bullet points, higher sodium outside, higher potassium inside. But when it does pump, it does not pump it equally. It pumps 3 sodium ions out of the cell for each cycle, but only 2 potassiums into the cell. So we are losing 3 positive charges but gaining 2. So each time it works, a net positive charge is lost. Okay, so again it becomes more negative on the inside. So let's look at this pictorially. On this chart, it shows the plasma membrane here with some channels as well as a pump. This is inside of the cell, that's outside of the cell. So inside the cell we have more potassium ions, these pyramid-shaped positive charges. Outside of the cell, the green plus charges are sodium. So a lot more sodium outside, as well as chloride, which are negative charge outside the cell. Then next we'll talk about those large protein ions. So here they've drawn it relatively bigger with negative charges, so there's more there are more of them inside the cell, but they cannot move across. Next one has to do with the sodium and potassium leak channels. They've only shown 2, but there are a lot more potassium leak channels than sodium. It's 25 times more permeable to potassium than sodium. So we have a lot more potassium leaking out compared to sodium coming in. The last one is our sodium potassium pump, which will pump 3 sodiums out for every 2 potassiums in each cycle, right? So we will lose a positive charge. So let's talk about 2 terms of when we change the membrane potential. So it's relative to the resting membrane potential, and we have to remember that resting membrane potential, the most common is -70. So if we become less negative, we call it depolarization, so going towards 0 and above. Okay, so it's a directional term. If you go below -70, so going from -70 to -90, that's called hyper. Hyper means more, it's becoming more polarized, further away from 0. Okay, so going up, whether it's go towards 0 or even above. So if it goes from -70 to -30, 0 plus 30 even, that's considered depolarization. While if it goes from that -70 millivolt of resting membrane potential down to minus 90, minus 100, it is called hyperpolarization. So we're going to talk about two distinct types of potential changes. One is called graded potential changes. Two things you want to remember, or a couple of things, actually maybe even three things. First thing is that it is very important graded, right? So it's proportional to the stimulus strength. So if we increase stimulus strength, so in this picture it shows these bars as stimulus strength, the higher the height of it, the bigger the strength. So you can see the change is increasing as the stimulus strength increases, right? That's very important. Next one is that they are short-lived and they dissipate quickly. That means short-lived, and they decay. So if you imagine if you draw a straight line here, that was a cell or a neuron or even the surface of something. If we create a graded potential at this spot, it just spreads slightly to the next view and then stops. Okay, so it decays on. Where in the same scenario, if you had that one line as the neuron, we create what's called an action potential on one spot. It's going to spread throughout the whole cell. It does not decay till it gets to the end of the line, and then it has to be transferred to the next cell. Okay, graded potentials can be either depolarizations or hyperpolarizations. So if we have in this picture, it's depolarization going from minus 70 towards the 0, but we can also have hyperpolarization of these membranes, which can cause graded potential because they're proportional to stimulus strength and they decay. Okay, these are the kind of potentials that are created in receptors. Okay, next we're going to talk about action potential. So action potential is a change in the membrane potential, again as previously, but this change is how we communicate. The cells, the nervous system especially communicate, but we also do this in our muscles too. Okay, a couple of things: they do not decay, right? So you produce it on one spot, it'll go throughout the entire length of the cell. There is something called threshold, so we have to reach threshold to have a stimulus or to have an action potential. Okay, and we'll talk about this in the next slide in a second. So once we do produce, so there are some sub-threshold stimulus will not produce an action potential, right? But when it's produced, it'll be exactly the same. Even if we increase the stimulus, we just will send different. The number of action potentials sent will be different, so will be increased. For example, I'm sure you've been able to distinguish pain, right? Sometimes it's just a little bit of pain, sometimes it's excruciating. The way that the nervous system conducts that impulse from our receptors is by sending a greater number of impulses when we have a greater pain. Okay, and at the receptors itself will be graded potential, but the neurons will be action potential that's going to be traveling. So threshold, so not all depolarization. It has to be depolarization, right? It has to go towards that negative, towards the 0 mark. So a threshold is the voltage at which AP stands for. I'm using that for action potential. So the voltage at which action potential is triggered. So this threshold is usually around 15 to 20 in the depolarization range of the resting membrane potential. So our neurons, the resting membrane potential is minus 55. The resting membrane potential is minus 70. The threshold is minus 55. So it's 15 from that. Once it reaches that, we will have activities that trigger the action potential. So just before that, let's talk about two sets of gates, or one set of gate and another gate. So we have two channels that are going to be playing the biggest role in the action potential in our neurons, right? It is a sodium channel and a potassium channel. Sodium channel is very interesting. It has two gates. Okay, there's two both voltage gated, so changes in membrane potential will trigger their opening or closing. We have an activation gate. This is closed at rest and it opens when we reach threshold. Okay, so it causes depolarization because sodium will move into the cell. Then when a certain voltage is reached, we're going to have the inactivation gate that's going to close, right? So normally the inactivation gate is open at rest, but a certain signal will cause this gate to close, which prevents more sodium from entering and also prevents or the impulse from traveling in different directions, which we'll talk about later. Potassium channel has only one voltage gated voltage sensitive gate. It is closed at rest. What's important to note, it's slow to open, it's also slow to close. Okay, so we're going to talk about action potential, the different things that happen and the three phases that are involved. So first, resting membrane potential. So these squiggly with a round head are our phospholipid, that's our plasma membrane. These green channels are going to be for sodium. The yellow one is for potassium. So you can see the sodium ions, potassium ions. So this is the inside of the cell. This is the outside of the cell on top. Okay, so at resting membrane potential, we have -70. We have more sodium outside, more potassium inside. So some sort of a trigger causes the membrane to become -70. So for example, in our neuromuscular junction, we talked about those cationic channels that opened when the acetylcholine attached, and we had sodium coming in, we had some potassium leaking out, but sodium came in, went from that -70 to -55. Sodium is positively charged, so when it comes into this negative environment, it's going to neutralize some of those negative charges, right? So this -55 is the trigger for that gate to open. Oh, let me also go back. So this is the activation gate, and this is the inactivation gate. So activation gate is closed at rest, inactivation gate is open. The potassium gate is closed at rest. This is the only gate. Okay, so at -55, we're going to have the activation gate opening. So you're going to see the channels open, right? So this phase is called depolarization. So it's becoming less negative. This happens because of sodium ions moving into the cell. So influx of sodium ions. The charge of the membrane goes from -55 all the way up to +30. Okay, and that's the end of depolarization phase. At this time, the sodium inactivation gate is triggered and it will close. Okay, by this time, potassium channels have opened. This gate has opened. Okay, so hopefully you might predict which way the potassium is going to move. There's a lot more inside, so they're going to diffuse out. So when we lose plus charges, inside is plus, so it was at +30 at the moment. So when we're losing plus charges, that membrane potential is going to be dropping. So it's going to drop, go down to 0, become negative. This is called repolarization. We're coming back to the -70 spot. Okay, so first phase is depolarization, next phase is repolarization. Depolarization happens because of influx or inward flow of sodium. That's depolarization, right? So it goes from -55, which is threshold, to +30. Repolarization happens because of potassium efflux. Potassium leaves the cell, so positive charges are leaving. We're going from +30, it goes all the way down to 0 and to up to -70 degrees, -70 millivolts. -70 millivolts is the trigger for potassium channels to close, but I mentioned in the previous slide that potassium gates are slow to open, they're also slow to close. So by the time they do close, we have a few more potassium leaking out. So we have the third phase, which is called hyperpolarization. It becomes more polarized, it becomes more negative because we lost more plus charges. Inside becomes more negative. It goes from minus 70 all the way down to minus 90. These are the three phases of action potential. Please be able to explain this for as an understanding of this chapter. Okay, this is a very important information from this chapter, right? And then we have all of them resetting. So potassium channels close, the sodium channel, the gates reset, the activation gate closes, inactivation opens. We have sodium and potassium being pumped to the normal places, and then we'll replace back our membrane potential to minus 70. So here's another view. So when we reach minus 55 threshold, sodium channels open, sodium will move into the cell, so neutralizing and going beyond, becoming plus 30. At plus 30, sodium channels close. By this time, potassium channels are open. So we have potassium leaving the cell, so loss of positive charges, it becomes repolarized. Once we reach the 70, we have the channels, that's a trigger for channels to close, but they're slow to close. So we have a few more potassium leaking out. We have hyperpolarization phase, which is because of extra potassium leaking out of the cell. Okay, that's our action potential, 3 phases of action potential. So we have something called absolute refractory period. This is a time when no stimulus will result in an action potential. So this is because of that sodium channel having those inactivation gates. Okay, because those inactivations gate are closed, they cannot be triggered. To reopen, right? So the idea is that any other impulse, so it went from that plus 30 all the way past that plus the minus 55, which would have been threshold normally for sodium channels to open. This prevents that, right? Otherwise it's just going to be confusing, especially when we talk about propagation shortly. Okay, this ensures that it's a one-way transmission of nerve impulses and also an all-or-one phenomenon. So basically it's going to be in there. So let's talk about propagation. This is when that impulse spreads throughout the cell. Okay, so say if we take a line, I'm going to ask us to imagine a line that's going across the screen. I created an action potential here. So we have sodium coming in during depolarization phase. Nothing's going to stop this sodium to diffusing on either side. Okay, so when sodium goes to this next segment and diffuses to that next segment, there from minus 70 it's going to go down to minus 55, and that will trigger the sodium channels to open here. So this will also create an action potential here and so on. So it keeps going. Okay, so it is why that's called self-propagating. So one, that creation of that opening of the sodium channels and depolarization will cause depolarization of adjacent spots. This is again very important why we don't want that impulse to go backwards either, right? So we have in the previous spot we would have those gates closed so they cannot reopen again. Otherwise you're going to have impulses going up and down the same neuron and confusing us. Okay, in unmyelinated fibers or non-myelinated fibers we have what I just described, which is from one spot to the next segment it triggers an action potential, while in myelinated it's going to be different. Okay, and I'll show you that in a second. So here is our unmyelinated fibers. So one spot, okay, will create an action potential in one spot, which will create an action potential along to the next spot. So it's continuous to one segment to the next. So action potential just goes along the axon. Whereas in a myelinated axon, it's called saltatory. Saltatory is skipping. So what happens? It only happens at the nodes of Ranvier. So at the nodes of Ranvier, an action potential happens. You can imagine there is a speed train, a bullet train that transmits those sodium ions from this spot to the next node of Ranvier. So we've skipped through all this space where there was all this length, where there was myelin sheath, right? So we didn't have to create an action potential here to here to here to here. It just went from one node to the next node to the next node. So it's called saltatory. This is why myelin speeds up those chemical impulses. Okay, so next we're going to talk about synapses. Synapses are two main types. This is the junction between cells, right? So between whether it's muscle cells or neurons, right? So first type is electrical synapse. These are basically gap junctions between the cells. So if electrical impulse or a change in charge happens on this side, it'll just be carried to the next side. Think of those depolarization during depolarization phase. We have sodium coming here, will be just zipped through to the next step, next cell, which will then hence reach threshold, and they will also have depolarization, right? So this is seen in cardiac muscles, in visceral smooth muscles, as well as neurons in our brain. While other cells, even including some neurons in our brain, use the other chemical synapse. We also came across this in our neuromuscular junction, right? So there is a chemical is released, and the other side will have the receptor to it, which will cause either a depolarization, so allowing, say, sodium or calcium to move into the cell, or it can cause hyperpolarization, say if this channel, this receptor is attached to a chloride channel or a potassium channel. So chloride is negatively charged, it wants to come into the cell, so when negative charges come into the cell, it's becoming hyperpolarized. If we open a potassium channel, potassium is positively charged, we have more inside the cell, they will want to leak out, hence it can cause hyperpolarization, right? So depending on what kind of channel is attached is what the result will be, whether it's depolarization going towards the threshold, and hence it's called excitatory. We'll talk about this next slide. Excitatory postsynaptic, so that synapse is that junction on the other side where it causes excitation going towards that threshold, right? So becoming less negative. This is caused by depolarization. If we cause hyperpolarization, we open the potassium channel, potassium will leak out, so it becomes more negative. We call it an inhibitory postsynaptic potential because it's going further away from that threshold, right? Further away. Same thing if we open the chloride channel. Now chloride wants to move in, but it's negatively charged, right? So moving in, negative charge is moving in, again will cause hyperpolarization. Okay, so this is a quick review of our nervous system, nervous tissue, and also potentials. I hope you found this this video useful. I will catch you in another video. Have a great day, everyone.